Namaste students, I am JV Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic determine the ability of microorganisms to degrade and ferment carbohydrate with the production of and acid and gas. So this lecture belongs to the downstream processing lab. This is the first session of this subject. The subject belongs to the BSc, BT, BSc Biotech. So in this experiment, I'm going to describe about the general principle of the experiment and some introduction also. So what is the general principle of the experiment? The carbohydrate fermentation test is used to determine whether the bacteria can ferment a specific carbohydrate or not. Carbohydrate fermentation patterns are useful in differentiating the bacterial groups, or suppose that there are different types of groups of bacteria from which we need to separate the fermenting bacteria and fermenting non-fermentating bacteria. So this experiment will be helpful for selecting the bacteria which have the capacity to ferment carbohydrate. This test is used for the presence of acid and gas produced from the carbohydrate fermentation. The media is used as the basal media, which contains single carbohydrate source, such as the glucose, lactose, sucrose, or any other carbohydrate. A pH indicator is also added, such as the enriched solution, bromochrysol purple, bromothiamol blue, or phenol red. So the change in the color will indicate that the pH is changed, and the pH change indicative of the pH change is an indicator that fermentation is being conducted by the bacteria. So this test is very helpful. So when microorganisms ferment carbohydrate on acid or gas with gas, then they produce gas. The production of acid. So when the acid is produced by the fermentation method, then when acid is produced, this acid will reduce the pH of the solution, pH of the culture media, which will detect it, which will be detected by the color change. We are adding some pH indicator, and that pH indicator will detect that the color has been changed and fermentation has been conducted by the particular kind of bacteria. So Durham tubes are also inserted inside the upside position, and the bubble is formed, which will indicate that the gas is also produced. So, what is the process of the fermentation? First, a carbohydrate will be given with the bacterial sample. The glycolysis process will be there, and some pH indicator will also be there in the culture media. And then aldehyde or ketone will be produced, and carbon dioxide will also be produced. The carbon dioxide production will be detected by the Dunham tubes bubble, and the acid and gas production will be detected by uh, change in the pH. So we go to the procedure. So what is the media which is required in this process? In this process, we need a fermentation broth media. So what is the composition of this media? 50 kg of protease peptone. Okay. It is will be required 10 gram for 1000 ml. Sodium chloride for the 1000 ml 5 gram. Beef extract 1 gram. Phenol red 7.2 ml of 0.25%. And carbohydrate source is also required, which is 10 gram. This is the basic composition of the media. Now go to the procedure. First, we need to prepare the carbohydrate fermentation broth. First of all, we need to weigh the all the media, which is the trypticase, soybean, sodium chloride, beef extract, phenol red, and whatever the pH indicator which can be added. We all these compositions are weighed in the weighing balance, and they are mixed in the distilled water. They are dissolved in the distilled water, and they are dissolved with heat. Then we add 0.5 to 1% of the desired carbohydrates. Whatever carbohydrate can be added. Then we insert Durham tubes inside the tube in the upright position. Okay, the upper portion will be inside the tubes. Then we autoclave the prepared test media at 121 degrees centigrade for the 15 minutes. When the process has been autoclaving has been completed, we use aminos, lactose, maltose, casein, sucrose, citrose, xylose, and carbohydrate source. Autoclave is 121 degrees centigrade for only 3 minutes as the carbohydrates are subject to breakdown of autoclave. Then we transfer sugar into the screw cap tubes or fermentation tubes and label them properly. So this is the first part of the media. Then we go to the inoculation of the bacterial culture. After that, we aseptically inoculate each labeled carbohydrate growth with test bacteria. Suppose that we have three samples. Suppose that we have different tubes and we have added different types of pH media, pH indicator, and various growth indicators. We have also added different tubes also. So we use these different tubes because we have different bacteria also. For different bacteria, we are using different uh, tubes here. So in the inoculation of the bacteria, we aseptically inoculate each level carbohydrate with test bacteria and culture with inoculated load. One control is also there, which has not any kind of bacteria. We incubate the tubes at 37 degrees centigrade for 18 to 24 hours. And we observe the reaction, what is going to happen in the tubes. So if we observe the tubes after 24 hours, then we observe the acid production and the gas production. How we observe acid production and the gas production? If there is a gas production and it is a positive reaction, then what is the indicator of the positive reactions of acid productions? After 1 day, after incubation, the media turns yellow as the indicator of the color change. And this is the indicator by the phenol rate indicator. This phenol rate indicator will turn into the yellow color. And fermentation of the carbohydrate will lead to the acid production, which caused drop in the pH. And after drop in the pH, the phenol rate will change its color into the yellow. And what is the indicator of the gas production? If gas production is found positive, what is the indicator of the gas production? There is a bubble formation in the Durham tubes. We have inserted some Durham tubes. If there is a bubble formation in the Durham tubes, then it will indicate that the gas is being formed by the bacteria. It's indicate the test is positive. So what is the use of this experiment? It is used for the determinants of the fermentation reaction of the microorganism, especially enteric bacilli and enterococcus. They are primarily used for the differentiation and presumptive identification of the gram-negative enteric bacillus and different another bacteria also. So I must share the screen here so that students can get aware about the acid and gas production and In, in this slide, it is an inoculated tube which where no bacteria is inoculated. In the second tube, there is a formation of yellow color. Okay, there is a formation of yellow color which indicate that the acid is produced in this experiment. So E. coli use this carbohydrate and it is convert the pH is lowered, and after lowering the pH the color of the media is changed from the original to yellow color. But one more thing, in this tube we also observing some bubble here in the Durham tubes here, here, here and here. We are comparing of all of these tubes. Only this tube is showing some bubble formation here, which indicate that in this tube bubble formation is indicative of gas production also. So in this tube gas production is also observed and acid production is also observed. In this tube only acid production is observed. There is not any kind of bubble in this tube. So it indicates that only visible gas, visible acid production is observed, and in this tube, acid and gas production both are observed visibly. So I hope that students will be better understand it. So in this experiment, I explained the acid and gas production by the fermentative bacteria. So I hope it will be helpful for the students. And, the questions which can be asked in this according to this lecture is that explain the process of the production of acid and gas production by the fermentation of the carbohydrates. Explain complete experiment on acid and gas production by the carbohydrate fermentative bacteria, and write the principle and procedure of the acid and gas production by the fermentative microorganism. So this lecture is over here. I hope it will be helpful. So if you have any questions, you can ask me. Thank you very much.